some work, and I've really enjoyed all of the talks so far. And I've also really enjoyed hearing about the different perspective that people have brought to what they are working on right now, like hearing about Bob Wright and his clinical practice in pediatrics, and then Rule with his experience in sound engineering, and how they shape that. So I was thinking, well, how does my background influence what we're working on right now? And really, I had a training in atmospheric chemistry, and that has really shaped on how we're moving forward with the bulk of my data. So while doing this atmospheric chemistry work, we really generated these really rich compositional pieces of data sets. We were doing this on a spatial network, high time resolution, and as I shifted from atmospheric chemistry to environmental health, it was really a bit perplexing and surprising that we just lost all of this chemical detail when it came to our exposure assessments, especially when we started working on the personal level for exposure assessments. And that's really what my group now seeks to do, is how can we bring back all of this rich chemical information that we know that we're exposed to, and then start to put that back on a personal scale. And so that's what I'll be talking about on airborne organic exposures. So a lot of my work, a lot of work that we've talked about here, have been able to characterize air pollutants, and we can do this really well with the available tool set. And this is where I've been finding where the issue is that we're lacking available tools that we can start looking at a range of different exposures at the personal level over this continuum of life stages. So if we'd like to measure air pollutants, we can outfit people with a backpack or some sort of a number of instruments that are fairly expensive that will measure a single type of pollutant. So we can measure PM mass concentration, we can measure number concentration, black carbon, perhaps collect it on a filter for offline measurements. And we can do this really well for maybe a day, a week. But going over longer term, it creates some challenges. So now thinking on how to move forward, we think about the potential caveats that we want to address. So the first is, how do we start getting at these age groups that are more challenging to outfit with our current tools, and think to move beyond, to really start getting at how can we start doing exposure assessment for the pregnant women when we know that exposure is critical, or the infants, the young children that are just not suitable for wearing these backpacks, really expensive toolkits that we worry that they would just drop and break thousands of dollars worth of instrumentation, or even these elderly populations, so that we know that they have been diagnosed. We're interested to see how treatments are affecting them, but they're just not feasible for carrying around these big toolkits. And also meeting resistance with clinicians in giving these expectations for what their patients are to be wearing to give that exposure information. And then the second caveat we see with the current tools is they're expensive. So we look at these real-time monitors, and they give this fabulous real-time data that someone can wear. But when we start scaling up for this type of exposomic type of analysis, and we want hundreds, thousands, tens of thousands of individuals to participate, we really need to start bringing down that cost price point. So that's what we've been working on doing. So in thinking how we can design this backpack, this really expensive toolkit that we give to people to look at personal exposure, we started asking multiple questions. So the first is, what is a desirable form factor that someone would actually want to wear to be able to characterize their exposure to the external panel of pollutants? What is an appropriate cost? Thinking about what a potential research budget can accommodate with NIH funding, and also potential citizen science type of engaging at the participatory level. What's the desirable compositional breadth that we want to incorporate, getting that comprehensive type of approach? And then what's an appropriate time resolution that we'd easily incorporate into our analyses when thinking about different health outcomes? So we took the standard right now, this method of active sampling, and when we talk about active sampling, this is the means of pulling air through some sort of filter material. So we have a battery that operates a pump that's pulling air through some sort of filter, and we thought, well, if this approach is creating things to be really expensive, bulky, how can we start to eliminate some of these components? So we said, well, let's throw out the battery, let's throw out the pump, and then just really focus on the material on which we are collecting our pollutants. So we turn to passive sampling. So solely that sorbent material, and relying then now on a diffusive type sampling to collect onto the sorbents. So one of the materials that we selected to use was a polydimethylsiloxane, or PDMS. And this is historically being used in a lot of other type of applications for passive sampling. And it is really fantastic at absorbing classic air pollutants that have been studied for a number of years. So things like volatile organic carbons, or VOCs, polycyclic aromatic hydrocarbons, PAHs, but it also gets us to get at an emerging class of compounds that haven't traditionally been measured in an air pollution perspective. Things like PCBs, pesticides, PBDEs, and also phthalates. The uptake of all of these compounds is proportional to the octanol water or octanol air coefficients, so we can theoretically predict the sorbent capacity of each one of these compounds within this material. So I mentioned before that this has been applied to a number of different applications. So cases solid phase microextraction, or SPME. This is traditionally used in analytical labs to look at different types of samples, water samples, urine samples, where you have this needle that's coated with a PDMS or some sort of other polymetric material. And then you have absorption of all of these different contaminants into that material that's then analyzed by a mass spectrometry approach, typically gas chromatography. So we can look for this range of contaminants in a thin film of coating into this thin layer on this SPME fiber. So we thought, well, we could just deploy some of these SPME fibers on people. One of the caveats, though, is that they're fairly expensive, so each one of these is about $200, and they're incredibly fragile. So you have this thin fused fiber that I know if I put it onto my 4-year-old, it would break within seconds. So really, we're trying to use this concept, but just make it more robust. So our solution to that was thinking about how can we now take a piece of glass bar, coat that with PDMS, and then now have a much more robust type of material that we're sampling onto. It's also reusable, and now we can start making this in a high throughput manner within the lab for wide deployments. So we fit this into a wearable type design. So we have two formats, either a wristband format, such as one that I'm wearing right now, or into a magnetic clip that can be fastened onto some garment or to some backpack. And within this sampler, we have this white casing that is made out of an inert material right here. So this is a PTFE virgin Teflon. We use this so we have some sort of casing to protect our sorbent. And within there, we put these PDMS bars that I described. So typically, we put 4 to 5 of those in there, depending on the study. And then we have now this chamber that can then be mounted in a wearable form and then given to participants to wear. So we're also thinking about how we're doing our analysis, because the throughput and feedback is important. So we start by giving participants these wearable devices. We have absorption over our known exposure period. They're then sent back to the lab for analysis. So when we're doing this sampling, we have a combination of gas and particle phase sampling, which is important to note. So we have diffusive gas phase sampling. We also have deposition of a particle phase on these sorbent bars. So after our analysis, we're interested in doing multiple different types of analyses. We'll spike with a known amount of deuterated standards, so that allows us to do a quantitative targeted analysis that we then analyze using a thermal desorption method. So we have an oven on the inlet of our gas chromatography mass spectrometer that then allows us to do a direct high throughput sample analysis. So in our case, we're using a Thermo Q Exactive GC that then affords us the luxury of then being able to do targeted analysis, suspect screening, and then also untargeted. So I'll just highlight some of these methods and then show you some of the results from our targeted and suspect screening data. So to give you the breadth of range of compounds that we're able to analyze, I mentioned before that we can do a range of different polycyclic aromatic hydrocarbons. We can also do various smoking-related compounds, such as nicotine and THC, a range of VOCs, PDBEs, phthalates, flame retardants, and then also a range of different pesticides, including pyrethroids. So quite a comprehensive list, which gets me really excited, but then we can also move on to a suspect screening approach. And here we take this workflow that's described here. So we have our GC acquisition from our Orbitrap. A subset of our samples, we then undergo a deconvolution and identification protocol using a thermo deconvolution plugin that then allows us to apply a stringent set of filtering methods that we can then use to create a targeted peak list. We're using MZMine to do this, and then we can then apply a list of additional filtering to remove blank features and then also any duplicates. So we've now applied this approach to a range of different studies to do exposure assessments. This map really gets me excited just to see the range that we're able to go on. We've been able to deploy in a number of different birth cohorts. So we have active sampling within South Africa in the VEMB study and also within another study within China outside of Beijing. And we're really excited to start partnering with a group at Dartmouth to start working with the New Hampshire birth cohorts. We've also been sampling other age groups, some adults and also children in various places across North America, and also now with IS Global, adults and also some women within India. We've been sampling then also at the other end of the spectrum with seniors in collaboration with the China CDC. So not only are we doing single measures of exposure, but we're also starting to do repeat measure of exposure to then get a longitudinal analysis. So one of these examples where we're doing repeat measures is within China. In the case of the Xinyi birth cohort, we're doing measurements in each trimester that we can then pair with different health outcomes that are being measured, such as ultrasound measures. And within China, I'll describe that repeated birth cohort a little bit more later. In addition to doing personal exposure, we can do stationary sampling. So in addition to the personal sampling we've done with our seniors within our China CDC cohort, we've also done some more sampling in other regions of China and much more extensive spatial monitoring within Montreal, Canada. So to highlight some of this data, because it's a rich data set that we look on, we started to consolidate here. I want to just highlight 3 different vignettes of results that we have. So the first is, how do these exposures vary by region? The second is, how do these exposures vary by time? And then finally, how do they vary across space in order to start compiling this exposure framework? So I'll just highlight some of these samples to give you a little background on the types of cohorts that we're working with in the different regions. So the first here is what we're working in the Limpopo region, which is in the eastern part of South Africa. This is a birth cohort that was originally started at Berkeley and then transitioned over to McGill in 2012, 2013. About 750 women originally were recruited. We're now working with 600 children that we're now sampling longitudinally. So we started at 5.5 years and now moving subsequently all the way until the age of 8 at 4 different time points. We've also been working with collaborators at the Karolinska and JSS Medical College to sample women in Mysore, India that have incredibly high levels of biomass exposure. So this is an existing cohort that they were working with about 10,000 individuals that have been diagnosed with chronic obstructive pulmonary disease or COPD at an incredibly young age, within their 30s, due to high levels of biomass. So we've been able to work with a subset of the women that are within this cohort. We've had some phenomenal collaboration with the China CDC in their BAPE study, where they've recruited 76 seniors to measure longitudinal exposures, measuring exposures every single month over a 5-month period. So now we can assemble this time course of exposures. And finally, most locally, I'll show you some exposure results from New Haven with some of our student sampling. So just as an overall perspective of the types of exposure results that we're seeing, I've grouped the different chemicals by chemical class and just shown these donut plots to highlight key features of the exposure. So this is the percentage combination contribution for each one of the exposure types. And you can see that by the different regions, there's some results that really stand out, and they're all quite different on their own regards. So obviously the first thing that jumps out to me is this really dark navy band for South Africa corresponding to phthalate exposures, and this really struck us as being an exposure that we weren't expecting. And we're seeing phthalates come up repeatedly over the course of all of our cohort groups, including India, China, and a very large percentage within New Haven with our Yale students. We also see a strong biomass signature from the India cohorts, which wasn't surprising given the contribution of biomass time that's being spent by the women that were sampled. So I'll just highlight some of these results here, focused on some organophosphates, some PAHs, and phthalates. So I'll start with the biomass signature. We expected, these are some pictures of the women within India. You can see that they were wearing the bands in proximity to their stove that they were cooking with. Not only are the women present, but they're also present with their children. So that's an extension that we'd really like to pursue further, is looking at these exposure levels and any health outcomes that manifest within these children populations. So I plotted here box plots for China, India, South Africa, and you can see that for all of these PAHs that have been shown here, we have elevated levels within India compared to the other locations. And we can now turn our attention to the phthalates. Again, similar types of plots with China, India, and South Africa shown for 4 different phthalates. And you can see that there is some level of correspondence between some of the phthalates, some higher levels that we see in China, and in other phthalates, higher levels that we see in South Africa. But really what I was surprised by here is the high level of phthalates that we were seeing in India. So we knew that they were burning wood. We had made the assumption that it was clean wood that they had procured for use. But when on further expansion after seeing these results, we saw that it was really finished wood products that they were combusting within their homes. So it was coated with a number of laminates, a number of coatings, and this is reflected in these elevated levels of phthalates that we were seeing here. So it's really not just this high PAH signature that we're seeing with the biomass, but it's also potentially a number of other endocrine disrupting components like the phthalates that could be contributing to the resultant effects, health effects that we're seeing. So South Africa birth cohort was started because of use of indoor pesticide spraying for high rates of malaria within the region. So we were expecting to see different levels of pesticides within this community, but just to give you of the prevalence of the number of exposures we were seeing across this cohort. So I've shown here levels for DDD, DDT, and then also peptidyl redoxylase. You can see that across most of the children that we sampled, we saw levels for each one of these. This is an example here of the spraying that goes on in this region. You can see that the children's toys are also present in proximity to all of these pesticides. So if we shift our attention to how do these exposures vary by time, this is an example, data taken from the collaborative study with the China CDC. The basic study design here was that we had seniors that were aged 60 to 69 years. They wore the wristband for 3 days, and they wore it 5 time points. They wore it September, October, November, December, January over this past year, and then we were able to then consolidate all that data to look at time trends. So to highlight the range of data that we were able to collect here, between our suspect screening data, we generated 611 different identified compounds, and that's shown here for a subset of those compounds, but over the course of the 76 individuals that participated within the study. I'll just pull up one compound here to show and highlight the range that we were seeing in terms of exposure levels. And you can see that with these box plots here, each one of these individuals has 3 defined repeat wristband measures. You can see that some people are explicitly consistently high, others are low. And so it's been really interesting now think about the variation, the intravariation, and then also interperson variation as well. Here's an example of a compound that we were slightly surprised to see within North America, or from a North American perspective, of dichlorvos, which is banned in the US, but still readily used within China. And this is used for mosquito control. So this is either sprayed within households, or there are sticks that are put under beds or different furniture to then volatilize for repellent of mosquitoes. Here's an example of results from one individual. You can see that over the course of the warmer period and as the weather cooled, there were decreases within exposure level as expected, and this is really then magnified as we look across the entire population. And so this is shown on a log scale to highlight the range of levels that we were seeing. And you can see that this drop temporally as the need for this insect repellent decreases. So something to highlight here as well is that in addition to doing our personal exposure monitoring, we were also placed some of these wristbands at an outdoor location. So this was at a governmental monitoring site. And you can see how the personal exposures differ from the outdoor exposures. And levels of the personal exposures are higher than the outdoor levels. And we see this consistently with a lot of the different compounds that we're measuring. So it's not sufficient just to assume that the outdoor levels are going to be indicative of a personal level exposure. So to highlight the range of the different concentrations that we were seeing, noting that this is on a log scale, I'll just show this one individual here. If we remove that log scale, just to show the range of different levels that we were seeing. So some other cases of temporal variance across compounds. I showed you dichlorvos as an insecticide, some seasonal use patterns decaying as the weather cooled. We also see the opposite trend with different other compounds, one example being limonene, used within a lot of cleaning products in addition to a number of other sources. But we assume that the increase with the holiday period and increased or decreased window opening then led to increased exposures of this particular compound. And again, noting the levels of personal exposure far exceed the levels that we were seeing at the outdoor sites. We're also able to measure compounds like triclosan. Given the use within a number of personal care products, we saw zero difference between across seasons. And then also compounds like BHT, also widely used. One example of a preservative. We do see some changes across season, but not pronounced across a number of the longitudinal time points that we were looking at. So what if we now consider exposure via space? So now we're moving away from the personal exposure, but placement across cities. And one case where we sampled this was across Montreal. This is in collaboration with Scott Weichenthal at McGill University, where we had 2 students, including Elizabeth Lynn, who is in the room here, place samplers at 50 locations across Montreal. And be able to then evaluate exposures at each one of these locations in the city. I've plotted here against population density, so you can see that there's some regions of the city where there's increased industrial activity in the north here, and you don't have as much residential communities. So also to note here that these sampling locations were done over the course of 2 weeks. So we're able to potentially then assemble time trends for each one of these exposure variables. We were able to do this for the range of different compounds, and I'm just highlighting 2 examples here of compounds that we didn't expect to see, one detect and then also be able to evaluate spatial variation for. So in the case here of fungicide, with applications for a different vegetable treatment applied to trees, also grass areas used as a wood protectant. And you can see that with the high levels shown in red, lower levels in purple, we do see spatial distributions, higher levels across this residential area here. And similarly with TCPP, a flame retardant used in a number of consumer products, foams, electronics, used for waterproofing as well. There's also strong spatial variation across these. So shown here is just an inverse weighted distance spatial plot here. Really the next step is moving into a land use regression map to then be able to map all of these different types of exposures and then consolidate these as a mixture across these spatial regions. So the next step, here's an example or an overview of the workflow that I mentioned previously for looking at suspect screening. We were able to then identify a number of different compounds. So going from over 6,000 chemical features that we're able to do with this just passive wristband approach. After deconvolution, identifying about 1,200 identified compounds, and then after further filtering, paring down to about 600. So then how do we interpret this? We do a number of this work in terms of integration with epidemiological studies, but a lot of this makes the wearable device pairs very well for community participatory research. And in doing this work, we're cognizant of the feedback to give to communities and being able to rapidly give them this feedback. So one aspect that we're interested in is assessing how does this mean in terms of communicating this risk and coming up with an automated platform to be able to screen these high number levels of components to be able to interpret what these exposures mean. So we've developed a software platform that we've affectionately called ChemCat, which is chemical classifications, that allows us to use available EPA databases to then screen these compounds that we identify. So we look at major and minor sources using identified sources through the EPA CPDAT database, and now we're also starting to then pair with predicted toxicities through the EPA CompTox dashboard. And I'll just highlight some of the ways that we're able to use these automated platforms to screen through our data in a rapid manner to then give feedback to communities. So here's an example of all the different sites that I mentioned to you from China, South Africa, India, and New Haven. And we're able to very rapidly pull out different categories that had been assigned to all of the chemicals that had been identified within CPDAT. So this ranges from food, consumer use, fragrance, manufacturing, pesticides, cleaning and washing, drugs, et cetera. So these are compounds that were flagged within this ChemCat platform and highlighted a number of different pesticides, just shown as an example, ones that I discussed previously, dichlorvos within China, the DDT compounds within South Africa, but it also highlighted some interesting compounds within New Haven that we weren't expecting, applied to different food products and also different applications to textiles. And it was really quite interesting to see that we were able to see these off-gas products from these products used within the New Haven area on individuals that wore the wristbands only for a short period. So we've also now started to apply this in terms of evaluating toxicity and using the CompTox platform. So here we took the top 5 ranking compounds with the highest predicted LD50 from CompTox, with triclosan at the top and dichlorvos following, and we're able to then screen in terms of compounds that people might be exposed to evaluate the implications of their exposures. We also now start to evaluate this in terms of a toxicity burden as a crude measure of potential risk. So we can then evaluate this by taking this predicted LD50 from the EPA databases, coupled with the measured TIC for each one of these compounds to then get a toxicity burden that allows us to re-rank the different exposures that we measure. And non-surprisingly, when we look at exposures that we found within China, we can see dichlorvos coming at the top in terms of the total predicted toxicity burden ranking, followed by BHP. We can do this for a range of different toxicity measures that are included with the CompTox platform. I've shown here an example of the different types of measures that are available for the top ranking compounds. We can also then start to rank this across the different exposures, really just to give us a flavor of different compounds that might be of concern for that population and help us to inform and rapidly inform the individuals within these communities. So in the last part of the talk, I want to just then also highlight some common questions that I have addressed with regard to personal samplers. So we've talked about some other different passive sampling techniques, including silicone wristbands. So I want to address how do different passive samplers differ from one another, and how do they compare? Another common question is, how does activity hand movement influence uptake rates on these samplers? And then finally, if we're thinking about wearing passive samplers in terms of exposure assessment, how long should I wear the samplers? And also, how can we start to think about moving from looking at a mass loading that's commonly reported for using these samplers into a volumetric concentration, so expressing these in terms of a picogram or nanogram amount per cubic meter. So commonly used passive samplers that have been reported within the literature, I've shown here these fresh air clips or wristband formats. We've talked about using these silicone wristbands as well, again, worn on the wrist. And also, there's been a number of studies that have reported use of a PDMS badge or a brooch formats. So all of these different sampler types are going to be capturing an inhalation type of exposure. But it's important to note that when we look at these silicone wristbands, because they have direct dermal contact, they also then have this combined exposure route. So it's a combination of an inhalation and also dermal type exposure dose. And that's a key feature to note as a difference. So I've summarized the different features for all of these different sampling approaches just to highlight where they are similar and where they start to deviate. So the first, as I mentioned previously, a comparison between an inhalation and dermal combined exposure compared to inhalation. The second key feature that is important to note as difference is the difference between an open-based sampling type of approach, like the silicone wristbands and the PDMS badge or brooch, compared to a sheltered design that I've described with this fresh air wristbands. Third, they use different amounts of sorbent material. And the amount of sorbent material is going to influence the capacity of those materials to uptake different environmental contaminants. And then finally is the analysis approach. All of these analysis approaches can be harmonized, and there's a move, some efforts to evaluate how the different analysis approaches deviate and how they can give differential results. But a combination of them will use either thermal desorption or solvent extraction, then coupled with a gas chromatography mass spectrometry. So I'll just highlight the influence of now movement and how it's impacted by the different designs. So I mentioned before these PDMS type badges or wristbands are very effective at sampling a range of different compounds. All of the sampling occurs through diffusion through some boundary layer. So if you have very vigorous hand movements and you're wearing the sampler on the wrist, we can then expect that that boundary layer here is going to be reduced. That's going to change the sampling profile for those different compounds. So I'll show you an example of some results that were done by Tom Hardner at Environment Canada back in 2003, but they're really good at indicating what the magnitude effect this will have on just our sampling profile. So here is looking at PCB 153. He evaluated what the concentrations are on a similar type of polymetric sorbent material over time, and then looked at the mass loading under the effect of wind and no winds. And he saw that winds enhance the uptake rate of this PCB. So it's important to start considering where we start placing these passive samplers in terms of what that means in terms of what we are measuring. So this can be analogous to stationary sampling if we have it located at an indoor location or a sheltered outdoor location, or in the context of a personal type sampling. So one of the motivations for the current design with this fresh air wristband that I've shown you some results for is how we can then use a sheltering type approach to minimize the impact of all of these type of movement approaches. So when Tom Hardner did his study with colleagues and evaluated the influence of PCB, a range of different PCBs, wind and no winds, he had deployed them in an enclosed sheltered sampler. And this is typically what's used within his GAPS network that have been deployed at multiple sites internationally to evaluate a range of different outdoor exposure levels. And he was able to then look at the differences within the mass loading on these different sorbent materials in a sheltered environment. And was able to see that as soon as you start sheltering your device and you minimize the impact of wind on uptake rates, you start to have a much more similar type of uptake for the no wind and the sheltered design. And this really motivated the design for this fresh air wristband that I showed you some results for. So this influence of housing chambers is an important factor to take into consideration when starting to compare different passive samplers that are available for use for exposure assessments. So the final comment to address is how long should I wear a sampler? And this is often discussed with many collaborators in terms of what fits the study design, but also what fits the design of the sampler itself. So some basic theory about how these samplers work. They sample in 3 different regimes. Initially, there's uptake of different components within a linear regime that then approaches to a curvilinear regime followed by equilibrium sampling. The uptake rate is constant within this linear phase, so really it's desirable to be wearing the sampler while it's still sampling within this linear regime. So it's important to understand how do different compounds vary in terms of their uptake within this linear regime, and how can we make sure that we're reducing our exposure assessment period so we don't go beyond this period for all different compounds that we're sampling. So I'll show you some results from some indoor lab testing that we did within our lab space within Yale, and also some real world testing we did in India to show you some differences in terms of the range of different wear periods that we gauged to be appropriate. So here are some results from our lab testing. We were looking at a range of different compounds onto the PDMS material that we're sampling within the fresh air wristbands. So this is a phthalate, a BDBE, and also a PAH. And you can see that they're linear for different durations of days. For the case of the PDB, or for the PAH, we're able to sample over a multi-day period before reaching an equilibrium. So this emphasizes the importance of considering where we're sampling and the duration that we should be deploying these samplers for, so we can interpret the data properly. Also shown here is then the rate of the different sampling and how it varies by different chemical within different classes. And I've shown here a range of different phthalates with the uptake rate then shown in terms of a picogram per hour amount. And you can see for a range of phthalates we're within this linear sampling regime, but the uptake does vary quite a bit over the range of different phthalates considered, and similarly we have the case for pHs. Now, if we wanted to make the extension in terms of converting from a mass loading that we have that's commonly reported for these passive samplers to a volume-based concentration, deploying these passive samplers in parallel with active samplers can then be used to then calculate an uptake rate and then re-express the exposure in terms of a concentration, in terms of a cubic meter amount. And then finally, I'll just highlight some of the results we did by deploying the samplers in India. And a big question was how long do we deploy these samplers in an incredibly polluted region? So these women were in very small living conditions with their wood stove burning within the background. And we knew that sampling for a multiple day period was just going to saturate our sampler very quickly. So we had the women wear 3 different wristbands simultaneously, and then they started to remove the wristbands after 30 minutes, 60-minute, and then a 90-minute period. So then we could start look at the concentrations that had been loaded on the wristbands over that time course. Shown here are results for phenanthrene and then also a phthalate, and we can then see what the mass loadings are over the course of different sampling durations. And we found that these sampling uptake rates vary by participant given on what their home environment was and the magnitude of their biomass levels that they were burning within their home, just to demonstrate the variability, and then also emphasizing the short period that we had to sample for in order to reach saturation within our sampler. So whenever going out to do these personal exposure studies within different regions with different levels of ambient concentrations, it's important to do samples like this to make sure that we understand the dynamics of the sampler. So to sum up, we know that from this talk and also previous talks that it's important to be able to comprehensively characterize these environmental exposures, and really to be able to do this effectively, we need new techniques that allow us to do this exposure assessment at a personal level. I hope I've been able to show you that there's a range of different passive sampler technologies that are available, and when coupled with high-resolution mass spectrometry, we can really be able to now start to characterize a range of different chemical exposures. And now that we've deployed these samplers at a range of different locations applications. We were able to see differences in different countries and also variation across time and space, which can then hopefully be now embedded within these exposomic type of epidemiological studies, as Dean Jones mentioned earlier this morning. And finally, the design of it really now enables as a platform to engage with community-based participatory research, as I like to call them, as an environmental fashion accessory to give the community information about what their exposures were using a low-cost format that's highly wearable, and then also thinking about how and when these results can be interpreted with available platforms. So I'd like to thank my research group at Yale, and then also a number of our collaborators at the China CDC, Karolinska, the JSS Medical College in Mysore, India, McGill, UC Berkeley, and the University of Victoria. I'm happy to answer questions.